2 to 4 small cans of aspartame sweetened diet soda produces anxiety, alters brain activity, and these effects are heritable. At least these were the findings of this study. Hi, I have a PhD in immunology and I want to dissect this research to evaluate if in reality diet soda causes anxiety. We're going to analyze this research paper, and although the title might sound complicated, we'll break it down in a simpler manner for all of us to understand. As we know, aspartame is an artificial sweetener widely used in light or diet products due to its sweet flavor and low calorie count. But there's been public concern about its safety, especially when used consistently. Now, in this study, a mouse model was used. Contrary to what you hear in social media, research on animal models presents advantages and it's considered a high-quality evidence, and it's also valuable to understand human illnesses. These mice were divided into various groups. A first control one supplied with regular water, and other 3 groups supplied with different dosages of sweetened water using aspartame. Mice were allowed to drink freely from this water. The doses were decided considering that human daily consumption is recommended to not exceed 50 milligrams per kilogram. However, due to physiological differences, doses were adjusted. They were adjusted to the mouse using a formula, and at the end, the doses corresponds to 14, 7, and 3.5% of the daily intake value in humans. In simple terms, this was done to maintain equivalency. And the experimental animals were tested for anxiety indicators. The first test that I actually have done in the lab is the open field test. It consists of a box or arena where mouse explore freely and their movement is tracked. Normal mice tend to explore the whole box and spend a significant time in the center, while anxious mice lean more towards the walls of the box as if they were avoiding a threat or a predator. This indicator is measured as time in the center, and researchers found that after 8 weeks of 7 and 50% aspartame intake, induced anxiogenic effects when compared to the controls, and this result was observed in male and female mice. Researchers also performed a second test called Elevated Plus Maze. They performed a variant that's shaped like a circle and consists of a closed and open arms. Anxious mice will spend more time in the closed arms and will frequent less the open arms. This test is considered a gold standard to evaluate anxiety, and they observed the same. Aspartame induced an anxiogenic effect, and when treated with diazepam, an anxiolytic compound, these effects were reverted, at least in the open field test. Researchers also evaluated GABA gene expression in the amygdala of aspartame-fed animals. GABA is an inhibitory neurotransmitter. Actually, the clinically used diazepam binds to the GABA receptor, increasing its activity to quote-unquote relax the nervous system, thus reducing anxiety. In contrast, we also have an excitatory neurotransmitter called glutamate. Researchers found changes in gene expression as shown in this heat map, where red indicates increased and blue indicates decreased gene expression. This was evaluated in the amygdala, which is a brain part that processes emotions. And in simple terms, the gene expression pattern suggested a rewiring in the GABA and glutamate signaling pathways that favor an anxious state. Finally, researchers mated the control and aspartame-supplied male mice with only regular water-supplied females to produce an F1 or first generation litter, and also produced a second generation or F2 mice. The offspring was supplied with regular water and anxiety was evaluated at week 8 of age. And surprisingly, F1 and F2 mice also presented anxiety behavior, and even though they were not directly exposed to aspartame. In conclusion, according to the authors, consumption of aspartame via drinking water at doses that are mouse equivalents of 7 to 15% of the human maximum daily intake, or 2 to 4 small cans of aspartame sweetened diet soda, produces anxiety in male and female mice. The anxiety is alleviated by diazepam. The aspartame exposure disrupts gene expression in the amygdala, a brain region associated with regulation of anxiety and fear response, and these effects are heritable along the paternal lineage. So, should we run in circles and avoid aspartame? What would happen if I'm planning to be a dad? Should I stop drinking diet soda? Well, not really. As horrifying as the study sounds, we're talking about an experiment where mice were fed with aspartame for 8 weeks. That's a long time considering their lifespan. A mouse lives around 1.5 to 2 years, so the equivalent in humans of 8 weeks in mice is about months to years. Also, the amount was equivalent to consuming about 2 to 4 cans of soda daily. I would like to think that regularly people aren't consuming that amount of soda for that amount of time consistently. This study is just a reminder that what we eat does have some effects on our physiology and potentially with our offspring. So we really need to practice moderation, especially with ultra-processed products. A problem I noticed with this study is that when you evaluate anxiety, the elevated MACE is a gold standard, and we only see the results for it on the first test, but not for diazepam or the test done on the F1 and F2 mice. And I couldn't really identify in the paper the reasons why that was that way, or if the results were not reported. If the latter is the case, maybe the results didn't adhere to the hypothesis, so this would mean that the results are skewed. And it's not a scientific practice to ignore results because they don't follow my beliefs. And I'm saying this with an educational intent. I do not know these authors or have contact with them, so I don't have certainty of a malpractice. What I do know is that scientific publishing requires rigorous peer review by experts in the field, so experts in the field considered this paper for publishing, despite the lack of those particular results. At the end of the day, this is just a single study, and further studies are necessary to confirm or contradict these results. Moderation is important when consuming these products, but it doesn't mean that one once in a while will hurt you. If you like this video, please give a like to help me with the algorithm, and see you in the next study.